industry, or we have Stephen Burgin from BASF as our industry partner, and we have from NC State, we have Dr. Josh Pierce. So start off, a little introduction. My name is Kathleen Denya. I'm the Director of Innovation Partnerships for the Plant Sciences Initiative. So I support industry partnerships for the PSI, which is an interdisciplinary research program at the university to bring in faculty from across the whole university to work on projects that are multidisciplinary and solve complex issues around agriculture. So yep, I support industry relations, and I also support entrepreneurship for the Plant Sciences Initiative. So thank you again for coming. So first of all, since some of you, maybe this is your first mashup, it is only the second mashup ever. So what is a mashup? My whole vision for this is trying to bring together industry and academia. So thinking about, we have a lot of great talent here at the university, having the students have opportunities to interact with industry, learn more about what's going on at the different companies that we partner with. And then from the academic side, learning more about what industry is up to as well. And then same, looking at new partnerships between industry and academia, hoping that this can be a platform for doing that. So we're going to have our seminar from 4 to 5, and then after that, we're going to go up to the Innovation Hub, which is on the 3rd floor. So the thought also for the mashup is that we want to give this as a platform for our industry partners who are members of the Innovation Hub to have a platform to talk to the local ag tech ecosystem. So I'm advertising this to be open to anybody in the local ag tech ecosystem to come, any companies that want to come, but the speakers are going to be the companies that are members of the Innovation Hub. So we're going to actually go up to the Innovation Hub so you can see it from 5 to 6 p.m. We have some snacks and some drinks, so hope you can go up there and we'll do a social mixer. These are the companies that are members of the Innovation Hub. We're really excited to announce that we have 2 new members of the Innovation Hub. One's Enview and the other is AgriPoint. So very excited to welcome them. And so we'll start also having them incorporated more into our different events that we have. So this is going to be our last mashup for this the spring, and we want everybody to go enjoy their summer. And so we're gonna take a break for summer, take a pause. And so please put on your calendars. We're gonna have 3 mashups in the fall. So September 12th, October 10th, and November 12th. I still have to figure out what the program is gonna be. So please, if you have ideas, if you wanna see a certain topic presented, or if you have a recommendation for a certain person, I'm open to your suggestions from NC State and from industry. So this is my email address. Feel free to contact me. All right. So our first presenter is going to be Stephen Burgin from BASF Seed Treatment. Stephen's actually an alum of the university. He got his bachelor's in biochemistry from the university. And so we're really excited to have him here today and talk about seed treatment and his path to through his career.
Thanks for having me, everybody. So like Kathleen said, I'm a NC State alum, 2002 biochemistry. Worked in the crop science department for a couple of years. And then I went to RTP, did a couple of years at a startup called Paradigm Genetics. They got bought out by a couple of different companies. Part of that went to Monsanto, changed. And then I went to BASF. So I've been with BASF for 18 years, a long time for me. I've held positions in the lab, so R&D is 10 years as a bench scientist. And I spent 5 years in our project management office learning how to kind of run the business, how to manage some of our larger projects, to now where I am as a global marketing manager for seed treatments, biologicals, and inoculants. So it's been quite a good journey. It all started here in Raleigh. It's been a very successful journey for me so far, and looking forward to retiring with BASF in the distant future. But like Kathleen said, so I work for Seed Treatments. So it's one of our divisions in agricultural products. So BASF is a pretty large company. We have multiple divisions. Agricultural products has a large presence here in RTP. We have 2 sites, both in one off of Davis Drive and one off of TW Alexander. Lots of different activities, but seed treatment is where I sit. Just want to go into a little bit of why agriculture is important to BASF and agriculture is important to the world. So a couple of global trends that are driving new challenges. The growing and aging world population. So I think the The world population is supposed to be 9 billion in not too distant future. How are we going to feed that? We have to double our food supply to meet that growing demand. And generally it's considered we're going to have fewer acres to grow that food on because of things like urbanization, right? So as our cities expand, we're starting to lose our arable farmland. We're not gaining very many acres as we grow older with the world. So how can we get more out of that acre as we continue to grow our world population? Energy demand and climate protection, that's a big one. Sustainability is a topic that's driving all industries, but especially agriculture, to do better. How can we do with less energy inputs? How can we do more with less water resources? So as climate change is going to impact, we see hotter climates in the US, we see hotter climates in Brazil, we're seeing wetter climates in some parts of the world. How do we continue to support the world population with food, with less arable land, and these challenges we're facing with different climate changes? Globalization, developing markets, we see Africa as a growing opportunity, but it's not a very commoditized country or region at this point in time. How can we continue to drive opportunities there? As we see the supply chain issues, COVID wreaked havoc on us. Like, so how is globalization going to change supply chain? How can we do better with the resources we have to get the products from a company like BASF to our customers, the farmers at the end of the day? And then I mentioned sustainability a little bit, but again, this is a very important topic for BASF, and it's different for everybody what sustainability is. But for us, is how can we make at the end of the day, make the land, the crops better when we leave it than when we start it with, right? So how can we continue to make the world a better place for everybody? And it's a little bit about, when you think about a crop protection product, it all starts with the seed. So you have companies who spend lots of resources, lots of R&D investment on making their seeds. So in the GMO seeds, you have the Bayers, Cortevas, the Syngentas, the BASFs. Very important. How do you protect that investment they put into the seed? The seed is generally considered the most expensive part of a farmer's investment every year, making that decision, what seed to buy and what seed to sell. So how do you protect that? The first thing you do to protect that is use a seed treatment. So this is what you're actually putting onto the seed. Then you have what goes into on the soil. So you have infrared applications, you have other types of applications in the soil. Then you have your foliar sprays on the crop later in the season. So seed treatment is really the first time you get to really have a protection of that investment. And that's where my role comes in, and then in other industries as well, other parts of the industry as well. But so what is a seed treatment? And so the seed treatment says application of a biological or a chemical crop protection product to the seeds prior to sowing. It provides protections from pests, diseases, and improves early growth vigor potentially. But at the end of the day, it's what gives you that protection once the seed is in the ground for that plant to come out of the ground, and in some cases grow to larger growth stages, right? So we have systemic chemistries we can put on the seed, and then they protect the plant for 30 to 40 days after germination until you can get those first foliar sprays on. And what's the benefits to the grower of seed treatment? We're going to protect that high value seed. We're going to allow for earlier season planting, right? So we can apply biological chemistries. You can continue to plant the seed earlier and earlier. The farmers are taking more risks these days. They're putting this seeds in the ground earlier. It's colder, it's wetter. How can seed treatments protect from fungal infection, right? How can seed treatments protect, from early pest diseases, or how can seed treatments do those types of applications? Those are the kind of things we're focusing on. Very important is uniform seeding and emergence. So as a farmer puts, these large farms, they're putting seeds in the ground, they're having doubles or they're missing, that's a yield loss, right? So putting a single seed in its place where it's supposed to go every time is very important. Seed treatments play a vital impact in that as well. And it can also reduce the need for other applications. So there are some seed treatments where we can skip the first fungicide spray because the seed treatment protects that plant long enough for it to get out of the ground, emerge, and be successful. So we can skip some fungicide sprays or even some insecticide sprays. So that's really important. Benefits to the environment, right? We have precise applications, lower use of product. If we can reduce the need for application of other crop protection products, we can own the entire field, and we can reduce potential off-target exposures. I think we've all heard about the bee populations and how we have lost use of neonics as a foliar application. In seed treatments, neonics are still allowed because we keep the product on the seed. We have a coating that protects it. It's a very important product and it works very well, but we don't have that ability to use neonics on top of the acre anymore. But in seed treatments, we have technologies to allow us to do that. And I just like to show this little graph, the benefits of a seed treatment. This goes towards sustainability, it goes towards stewardship and all those type of things. But if you think about the area of a chemistry on a whole treatment acre, you're approximately 10,000 meters square. If you go to an in-furrow application, it's reduced to 500 square meters. And then as a seed treatment, we're at 50 square meters on average for an application of a chemistry. It just really shows how we can have that precise application and still get the benefit that we need from the chemistry until the plant is established. And again, we have those fuller applications that are still needed later in the year. And at BASF, we like to think ourselves as pushing the boundaries of seed technologies, researching and looking for new chemistries. I think a new class of chemistry for seed treatments hasn't been found in years. There's a couple that are exciting and potentially coming to the market in later this decade, early 2030s. But that's really important. Investing money to find new technologies that can support crop protection and seed treatments in general is really important for me. Combining different technologies. So you think about synthetic chemistries and biologicals. That's a hot topic right now. How can we continue to drive that and drive value, bring value to the customer, bring value to the farmer, reduce our synthetic chemistry load, but still have the same protection on the acre? Looking at other plant benefits. A lot of times you hear about putting a biological on the acre and we get a given yield boost at the end of the year. But we're increasing the root mass. We're increasing the plant weight at the end of the day. Can I go towards silage? Can we protect more secure more carbon in the soil? These are all important things that a small application of seed treatment can bring value to. So it's really intriguing to see how these things work together. Within BASF, we have scientists around the world, right, in all different industries that we can leverage on. We can go to questions for real synthetic chemistry or biologicals or coatings. We have a vast pool of technology experts that we can really work together to drive towards a final product. And providing answers to just difficult challenges. The customers come to us every day. We have a challenge with this pest, or we have a challenge with this application. How can we devote our resources as a company to help solve these challenges? And at the end of the day, make their job easier. That is our goal. And if you think about, the performance aspect, the portfolio of a seed treatment, these are things you can put on a seed and generally go on every seed. A fungicide, an insecticide, an nematicide, a biological. If you're thinking about a pea and lentil soy, an inoculant, and a coating. So we're putting, 3, 4, 5, 6, 7 different products on, and most of the time it's multiple fungicides and multiple insecticides. So how can we really drive this value, and how can we tie all these things together as a seed treatment and as a crop protection company? And then thinking about the areas that we focus on as a company, we focus on corn, focus on soybean, wheat, canola, cotton, and barley. We do have other crops, but for us, these are the core crops that we like to focus on. This is the areas that we spend most of our research on and investments in. But there are other opportunities in other crops that we do have interest in and have plays in globally. These are the major ones. So now I'm going to get into part of the discussion where my colleague, who's unfortunately sick, is a little bit more of the scientific part. So I'm going to try to cover it, but I'll do my best. So just bear with me. So when you think about solutions on the seed, one of the first things we have to think about is the plant pathology, the biology, and the efficacy. So BASF, we take this very seriously. We do fungal screening. We screen thousands of different chemistry molecules and different biologicals. So biologists I can speak about. We screened over 150 different biologicals over the past couple of years for an ability to go onto the seed. Big challenges there, right? Most biologicals don't last on the seed. If you think about corn, we have to have a biological we can apply on day 1, say January 1st, 2022. It has to be effective on January 1, 2024, right? It has to survive on seed for 2 years. These are some big challenges. We looked at multiple species across key row crops. Again, row crops are our focus, but how do we really see which of these biological species or which of these different chemistry classes are going to work, bring the best benefit, and go together at the same time? And then we also focus on support for our LCM. So we have products in the market already. How can we continue to grow those products? How can we make them new? How can we refresh them, make them better? And how can we bring new innovations in? So find those new classes of chemistry that'll work as a seed treatment or another crop protection product. It takes time, I think, to have a new crop protection product that's about 12 to 13 years. And usually right now it's around a $250 million investment. So these things we have to be very certain of when we enter into them. We have to have the regulatory issues that we have challenges with, different regulatory environments across the globe. The US is totally different than Europe, which is totally different from South America. We want to bring a product that reaches all those regions. It's a lot of work, it's a lot of effort, it's a lot of intensity, but it's something that we feel we do pretty well. Another thing we think about, and this is one of the big focuses for me as this marketing for biologists and inoculant, is nematology. So nematodes are a huge pressure on row crops, fruits and veggie crops, ornamentals, everywhere. But oftentimes it's not known, it's not understood that something that you can't see is having such a yield drag on the product. So nematicides are a huge product for us. We have biological nematicides, we have synthetic chemistry nematicides. But how are we have a platform we're screening for new activity. We're here, so biologicals and chemistries. We have work done in RTP, we have work done in our location, our headquarters in Germany in Ludwigshafen, screening thousands of new molecules, hundreds of new biologicals to see how we can put this together with our current packages, or even create brand new packages to battle nematodes. One of the big challenges is, if you think about North America, the market's pretty mature. Almost every corn acre has an nematicide on it. Almost every soil acre has an nematicide on it. But if you think about South America, the big country, Brazil, for the longest time, the farmers didn't want to discuss that they had nematodes. If you had nematodes, the value of their acre dropped significantly. That's changed now because they understand the nematodes are having such a huge yield drag that it's important to take care of, right? But again, how do we take care with the synthetic chemistry they're not used to? Or how do we take care with the biological chemistry that may have, inconsistencies around? Again, that's where we spend a lot of our time and resources to make sure we have a consistent product, a safe product to use for the environment and for the crop and the land. All right. Let's see what else. And let's see, benchtop to higher tier greenhouse testing. So for us, everything starts in our greenhouse. Like we do an initial screen. We run it in the greenhouse, and then if we see promising results, we take it to field. We usually run field trials over multiple years once we have a positive hit. If it has a positive hit after multiple years, we take it to our other teams within BASF. How can we take this as a new product? So everything for us usually starts in the greenhouse, but it goes to the field as quickly as possible. And that could be the fields in North America, it could be South America, it could be Europe. Plant physiology here, another important part. How is our chemistry really affecting the plant? Is it impacting the germination? Is it impacting the growth and the vigor? Is it impacting the yield? These are things that we can't have, right? We want our chemistries and our biologicals to have a positive impact. At minimum, we won't have a neutral impact. So if we put a chemistry on the seed and it delays germination by a week, that's bad, right? If we put a chemistry on seed and we have a 50% germination rate, that's 50% of that farmer's, value they put into their bag of seed. So we want to have a 100% germination rate all the time. That takes a lot of testing, right? So those are something we take very important. We evaluate, the growth benefits of our chemistry. We evaluate, is a synthetic chemistry or biological the right path to market? Oftentimes we're seeing it's both. These hybrid approaches are what's important for us. The yield enhancement, at the end of the day, that's what the farmer wants. He wants his acre to produce more yield at the end of the day, but he also wants his acre to look just as beautiful as his competitors, right? So again, if we're putting a technology on the seed, it's planted, his buddy down the road is planting a different technology, and his technology is coming up green, uniform, and this guy's coming up, a little bit slower, spotty areas. That's not good, right? At the end of the day, the yield might be the same, but the picture that the field has is vastly important to farmers. Yeah. Seed safety. So this is one that usually takes for our seed treatment the longest time. Once we have a product that we feel is vital for us, we have to make sure that the quality assurance that we've done doesn't have any negative impact on the seed. So this can be anything from a new formulation of the existing chemistry to a biological to a brand new chemistry. What we put on the seed has to have a positive benefit at the end of the day. We do this for everything from coatings to biologicals to synthetic chemistries. And this is where we spend probably the longest time outside of regulatory practices in our investments. It takes multiple years, or again, once the corn has to survive for 2 years, 2-year seed safety testing for every product we put. If we change a single product out of one of those 7 or 8 we put on the seed, we have to go through a 2-year seed safety protocol again for corn, 1 year for soy. So again, making sure that what we put on the seed and how it interacts with everything else and how it interacts with the seed itself is vastly important. We cannot damage that customer's yield potential. Then application technology. I think I mentioned this a little bit when we talked about plantability, making sure we have a single seed and a single hole with the right spacing every time. Another big thing is flowability. So making sure the seed doesn't clog up the farmer's machinery. At the end of the day, that's their worst nightmare. They have a 3-hour window to get out there and plant. They load their seed and there's a clog. They have to spend that 3-hour window cleaning out their equipment, cursing us or whoever they bought the seed from, right? And then putting it back in and hoping that it works. So plantability and flowability are really important. Visual and physical characteristics, again, just like what the field looks like, it's just as important what the seed looks like. So when we sell a chemistry to some of our customers, they want it to be beautiful. They want it to be a vibrant green or a vibrant red. They want it to be shiny. They want it to be uniform, right? So making sure that we can apply, again, 7 or 8 different chemistries and a biological and a coating to a seed that's run through a processor at who knows rate faster than you can see is important. And if we have a challenge in the seed processing or at the planting, we hear about it, right? We can't slow it down. Worker safety and stewardship. For North America, we try not to use dry powders, right? You think about an industrial seed treatment, big facility treating thousands of tons of seed, the dry powder gets in the air, inhalation issues. So we have liquid technologies that we really like to focus on globally because we take worker safety very important. We don't want to have any human health concerns. We don't want to have any regulatory issues. We don't want to shut down the seed processors because there's an issue. And then again, this applies for everything from our chemistries and biologicals to our colorants and coatings. So I ran through that quickly. I'm sure I didn't do it the right justice as my colleague would have done. But just want to end up saying that, you know, seed treatments is an exciting part of agriculture. A lot of times people don't think about it. They think, hey, here's your seed, what's next? With biologicals increasing, with chemistries increasing in importance, with loss of chemistries in Europe, loss of chemistries in North America and South America from a regulatory standpoint, seed treatments are going to grow in their value. How we can apply these to the seed and bring that value back to the farmer. We're going to see the landscape further evolve as we improve our discovery opportunities and our platform performances. So as we continue to grow with new technologies, with AI, we're going to find new opportunities, new chemistries that we can put on the seed. But it's going to take time to do all the testing to make sure that we don't have any negative impact to the seed. Yeah, so at the end, we strive to develop safe and sustainable and complementary technologies to meet our customers' need. At the end of the day, that's what we're for. We want to make our customers happy. We want to make our customers profitable. And it takes a lot of time, a lot of effort. And it's something that we pride ourselves on being one of the best in the industry. So hopefully that was of some value to you guys. Happy to answer some questions. If I don't know the answers, I have one colleague here who can help me out. But I can always go back to our experts, you know, in the R&D center to say, hey, these are the questions I've been asked. How can we get some answers to them? Yes.
More of a business related question now.
I might be able to answer that one.
So when you, if a farmer didn't want to purchase an entire bundle of what you had, can you order a la carte?
Yeah. Yeah.
So seed treat.
Yeah. So if you think about corn, most of corn is industrially seed treated in the U.S. and it's controlled by Bayer and Corteva. They're going to pretty much tell you what they want you to put on the seed, right? But you can have your seed treated individually, and you can go a la carte. The benefit from sticking with a completed offer is we're going to provide you a guarantee that our chemistries work together and it's not going to damage your seed, right? So if you buy a fungicide and you put on a biological, or you put on a mycorrhiza fungus, guess what? That fungicide is probably going to kill that fungus, right? So there is value from buying the total completed package. There's a value to us as a company, but there's a value to the customer at the end of the day. But a la carte certainly is an option. We see that a lot more in distribution channels. When you think about the soy market, farmers can go and ask for certain products by name. They also given, they take a lot of advice from their agronomic, their extensions offices as well. So we do work with our extensions offices to find out what the challenges are, what the challenges the customers are having, and really how we can provide the best solution. So, yeah. Yes.
Okay, this is a very basic question, but when you say biologicals, could you expand on that? Is that like plant hormones or what kind of?
Yeah. So when you think about biologicals, there's three categories. There's three main categories. You got biofertilizers, you got biopesticides, and you got, what's the third one? Bionematicides, right? So fertilizers, pesticides, and biostimulants, that's the third one. Nematicides usually go on to biopesticides. And these can be anything from microbes to peptides to small chemistries, right? So it's really depending upon what the need is. RNAi is a big topic now going in as a biological. It's grouped into usually a biostimulant or a biofertilizer category. For BASF, when we talk biologicals, we're mainly talking about microbials. So we have bacillus species, we have different species of mycorrhiza, we have different species of inoculants, bradyrhizobium, right? That's what we think of, generally we think of biologicals, but of course peptides are playing a bigger and bigger role. As a seed treatment, again, we run into challenges with stability. So you put a peptide on the seed, you put that seed into the dirt, it's not going to last too long. It's certainly not going to last 2 years. So we think about those things, what works from a biological standpoint, how we can stabilize it, and how we can get it onto the seed, get it to the customer, and provide that full benefit.
For RNAi, do you use double-stranded RNA then first?
That's for a seed treatment, it's really relatively new. It's usually a single strand at this point in time. Double-stranded is more stable, but we don't have any applications for it as a seed treatment. Double-stranded RNAi is been around from a foliar standpoint for a while. Hasn't taken off. That was a big product when I was in the lab 12 years ago. And most of those things, they take time, right? It's tricky. So again, stability is a big issue. Double-stranded gives you that extra stability, but it's still not where we need to be. And usually that's a single target, right? So finding a broader application for RNAi is challenging, but it is an area of interest. Yes.
People commonly go looking for microbial biologicals, like soils or waters.
Go take a plot.
Anywhere.
Yeah. I mean,
Strategies.
yeah. So when you think about spraying in biologicals, I mean, the first thing is you have to be considerate of where it's coming from. So the Nagoya protocol is vastly important. So can you take biological from point A to point B? That has to be considered. Most of these biologicals are isolated from soils as a seed treatment. From different areas. Most of them have been sitting in a bio library for years and years and years. There are new discoveries, but they're still coming generally from field plots. Something looks interesting, they dig it up, they figure out, they characterize it, they try to tease it down to one or two. Sometimes they can't. If you have a consortium of bacteria, it's a lot harder to sell, right? It does something, but how can you explain from a scientific point of view what's doing what and how it does it and how it's consistent across the time? So yeah, most of them come from soil, but there are other ways. Out of time.
Maybe one more question?
Do you have like different layers that you like apply to the seeds, or will it all be mixed in one formulation and then the seed will be basically coated with it?
It really depends. So there's inline applications which is a coat like one layer, then a layer, then a layer, then a layer. But there's also industrial seed treatments are generally more of a mix everything together. which again, takes a lot of work to make sure when you mix it together, it all works and does what it's supposed to do. And then a final coating for your application, right? So a final coating to keep the AI on and to make it beautiful and shiny and pearlescent and that thing. So there's both applications. There's both ways to putting a seed treatment on.
How many like crop seeds do you treat at BASF? Is it like just like the most common for the like region, like the US probably has a lot of corn and soy?
I mean, so corn, obviously corn, soy, those are the biggest, canola, barley. We have fruits and vegetables, so any fruit or vegetable seed you can imagine, we have a fruit and vegetable business, so we treat those seeds as well. It's really the full gamut. So there's obviously as a company, we're in it to provide a product that provides a benefit back to the customer and back to us. So we're not doing every seed, but I would be surprised if there's a seed that is economically important that we don't treat. But the big ones are corn, soy, cotton, canola, wheat, barley. Those are the main ones, yeah.
Well, let's thank Stephen for his excellent. All right. So next, our speaker from NC State is going to be Dr. Josh Pierce. And Josh Pierce, he is a Howard J. Schaefer Distinguished Professor of Chemistry in the Department of Chemistry over in the College of Science. So really pleased to have him come and talk to us today. Over.
Got to figure out how to use the PC again. All right. Yeah, hello everyone. Thank you for having me. It's fun to be in a different neck of the campus for a little bit and talk to some new people about what we do. I'm wearing a couple of hats today, and so I took the mashup theme to an extreme, and I think my slides also represent a mashup of several different things, starting with just very briefly who I am, followed by a concept that the slide suggests here called molecular biomimicry. Computer is not happy. There we go. Okay. Which is a theme and a topic that I think resonates both with my own research program, but also one of the key platforms that we're imagining for the Integrative Sciences building that's over on North Campus. Well, going to be over on North Campus. And finally, just closing with a little bit about that building and the corresponding initiative and opportunities for this broader initiative and building to interact with it. And so that's sort of where we're headed here. Very briefly about me, I was born and raised in Pennsylvania, northwestern Pennsylvania, in the middle of nowhere on an 80-acre old farm. Spent my undergrad and graduate studies at Pitt in chemistry. Went to the Scripps Research Institute in San Diego in La Jolla, and subsequently started my independent career here. You know, I was a pre-med, you know, we talk about undergrads coming through, you know, loving health and related things, fell in love with chemistry, really applied to graduate schools and went to Pitt. I joined the lab of this guy, Peter Whiff, in 2003. One of the more interesting things I worked on during that time was the synthesis of this little molecule called JP4039. It's actually a commercially available molecule. You can buy it from Sigma, but it's a radiation protection agent, and so it actually is able to protect animals from the radiation, and so you can give it to monkeys and protect them from radiation. And so the Department of Defense is actually still working with those compounds in a variety of different capacities. When I went to my postdoc, I really transitioned to think about antibiotics, and this molecule you see on the screen here is an antibiotic called vancomycin. If you have ever been with a very severe infection, you may have been treated with vancomycin in the hospital. You probably weren't in good shape if that happened, but it probably saved your life. And so it's been a very important molecule for history. And so, you know, with that, I started here in 2012. And, you know, I think as noted in terms of, you know, BASF's motto of "we create chemistry" is, you know, brilliant when you're a chemist because you have a whole company that just goes around saying that all the time. But, you know, I think that, you know, when you think about chemistry and you think about people that make molecules and study molecules, a lot of times your training really interfaces with human health. And the driver for that is often that NIH is paying the bills of your mentors. And so for me specifically, I spent all of my training thinking about how we can use molecules to affect human health. But it turns out those same chemistries and those oftentimes those same molecules can be really important for all kinds of different things, including agrochemicals and plant health and plant growth and all these related things. And so that's going to be a theme we come back to in a little bit. And so the bottom line is, how do we think about this question of whatever your application area is for these new chemistries? How do we create new ones to make these molecular solutions for all of these societal grand challenges? Whether that's molecules that are on seed treatments, whether that's molecules that are curing humans of infectious disease or cancer, or whether that's molecules that are part of solar cells for the, making green energy, right? All of that requires the same fundamental concepts of how do we access new chemistry to do new things with it, right? The question of how we translate that is the next stage of that. But up front, we need to have ways to make new stuff. And so quite literally, the molecules that we think about in all of this, whether these molecules that are on seeds or whether the ones that affect health and energy, they have literally fundamentally changed our world. The invention of penicillin to treat infections changed the world's population dramatically. Our ability to impact the growth of plants and the health of plants and animals dramatically supports our population growth by providing the food needed to survive and live. And there's a book called Napoleon's Buttons. If you haven't seen it, it's 17 molecules that changed the world. And it has all these fascinating stories about how single molecular entities changed the course of history, right? And so, the Dutch used to be in control of Manhattan, right? And New York, right? And, it became New York once it was sold to the British, but it was sold to the British because the British wanted a tiny little island in the Spice Islands it was the major producer of nutmeg at the time. And it turns out nutmeg has a little molecule in it called isoeugenol, which is a very strong pest retardant, and at the time was thought to, and to some extent could fend off the plague. And so, it was a huge thing to be able to secure nutmeg, so much so that they sold New Amsterdam at the time to the British to become New York, right? And so one molecule ultimately is sort of the root of all of that. There's lots of other stories in that book, you can check it out. But regardless, the moral of the story is if we can make new chemistries and molecules, we can impact the course of history, whether that's by addressing these needs for agriculture solutions by 2050 or any other thing you might do with these. And as a chemist, we love this particular table, the periodic table. It's got all these elements on them, and we spend our time thinking about how we connect these elements in new ways to make these new chemistries and these new molecules. And those then lead to all of these products, as was mentioned in the last talk, right? And some of these are the biologics, the proteins, the enzymes, the microbes, down to the peptides and DNA and RNA. Others are small molecules, like you may see in synthetic polymers or drug molecules, right? The molecule aspirin is shown there to give you some perspective of the different scales, right? Some of these are natural, some of them are synthetic, but they're all made from the same elements, and the chemistry that's used to make them, whether it's the microbe out there in that soil or the chemists over in Dabney Hall, is the same fundamental chemistry, right? And so that's a message that I really, as a chemist, want to drive home, that biologics and synthetic molecules are formed of the same things and make the same things. And in fact, these molecules are not necessarily all that different depending on what we're talking about. There are synthetic molecules that are, very, very safe and very, very effective. And the most potent poisons we know are biologics, right? There are molecules made by marine organisms that, a few molecules will kill a grown man, right? They're natural compounds. That does not make them safe, right? And so those are very different concepts. And, so just to think about that as we go. As noted, I spent my career coming up through a drug discovery environment where we think about making molecules for drugs and really being inspired by nature to think about the starting points for how we do that. And so, we think about identifying these new molecules or chemistries, as we're referred to, that can do this new stuff. As noted, those same things are relevant for all these other applications. We're excited here at NC State to start thinking about how we can create those bridges and connections to do that in a more robust and meaningful way as we develop these programs and hire more people to think about doing things like me. And so nature produces many of these compounds. And in fact, many of the microbe biologics you might see in the soil are actually making thousands of small molecules that look like the molecules that synthetic chemists are again making in chemistry labs, right? And so, thinking about that interface of microbes and molecules is actually a whole other area where you can not only think about them separately, but you can think about them together, right? And so how you can leverage that. And so as I noted, I apologize for the PC to Mac issues here. This came from a Mac, and so we've got formatting text problems. The font in PowerPoint on a Mac fundamentally does not exist on a PC. And so it automatically just applies another font with a slightly different size. It's like 0.25 different, and then it doesn't fit in your text boxes anymore. It's fun. So anyhow, just my message is that synthetic versus natural doesn't mean anything. You need to actually do the studies and tests to understand that. And so these natural products, they're what we spend our time in our lab thinking about. They're molecules that nature makes. It could be trees making them, microbes in the soil making them. There could be marine organisms in the depth of the ocean making them. They all come from these different sources. We use those molecules as new starting points to think about creating new ways of making them and then thinking about new ways of using them. And that's a holistic sort of cycle. And as sort of along these lines, nature is this molecular factory, right? If you took a scoop of soil out there right now, it's full of these microbes that are just they're spending huge amounts of energy in their world, huge amounts of precious energy to make these libraries of molecules that could be defense molecules, they could be beneficial, they could be synergistic with the plants that they're being produced by or on. But regardless, there's these multi-step chemical syntheses happening within these microbes that are producing these molecules. And those are new oftentimes to the world in terms of when we discover them, they may be unknown. And so when we think about molecular biomimicry and what we mean by that in my world, what we mean is how do we think about taking inspiration from nature and mimicking it, either by making exactly what nature makes ourselves, or by being inspired by the things that nature makes and trying to do the same thing, but with something different, right? And so those are going to be brief topics to introduce here. And so one of the ways we can do that is by mimicking the form of nature. And so as I noted, nature is this molecular factory. Inside these microbes, there's all these enzymes, nature's machinery to make molecules. And these enzymes literally make chemical bonds, right? That's what enzymes do. They're proteins that do things, right? And so it takes two molecules and puts them together to make a new molecule. And these enzymes are in these conveyor belt sort of sequences. They pass molecules from one enzyme to another, and you go from simple stuff to complex stuff, right? And like I said, that's happening right now out there. It's happening right now within us. The microbes in our gut are making all kinds of molecules that are doing all kinds of things, largely that we don't understand at all, right? And so lots of chemistry happening. And so one way we can mimic biology is to say, okay, nature does that with all those complicated enzymes. Can we develop tools in a chemistry lab, maybe with metals or other catalysts, to mimic nature's processes to make those kind of things, but maybe do it simpler or faster or cheaper, right? And so that's one way we can mimic nature. And NC State is investing heavily in a space in collaboration between chemical engineering and chemistry and biochemistry to think about how we can accelerate that process of mimicking nature. And so through the use of AI and machine learning and robotics, how can we take these processes and accelerate our ability to evaluate their potential, right? And then ultimately interface that with the ability to actually explore the effect of those molecules downstream in a closed loop, right? And so the ultimate goal is to have a self-driving lab that can conceptualize, synthesize, purify, and biologically assess, whether that's for an agriculture purpose or for a human health purpose, in a self-optimizing loop. I joke in these kind of meetings that if we want to have a Ferrari at the end of the day, we have a bicycle, right? And so in terms of our ability to do this, and I could, talk over a stack upstairs about sort of where those hurdles are, but that's the vision. So that's how we can mimic the form of biology. But we can also think about mimicking function, right? And so when humans figured out how to fly, we didn't strap wings onto ourselves and flap them. Well, I'm sure lots of people did that, but it wasn't successful, right? We didn't build a bird, we built an airplane. And so we mimic the function of nature, but not the form of nature itself, right? And so we can think about that in terms of molecules as well. Nature, those microbes out there, the microbes within us or any other biological system has a very limited set of starting materials. And so the natural world has, your standard amino acids, it's got a number of other common building blocks, but it's pretty limited. Whereas if you open up a modern chemical catalog, you'll have this giant catalog with, hundreds and hundreds of thousands of starting materials, right? Nature sometimes builds things for their function, but the complexity of those things can sometimes far outpace what is necessary to achieve that function. It just did it with what it had, right? And so if you mix two complicated things together and make something great, that doesn't mean it has to be that complicated. It just means it, was what you made the first time. And so we can also think about mimicking function and thinking about how we can navigate that by simplifying these chemistries, but still keeping that important effect, whether that's an agrochemical or whether that's a drug molecule. And so, I'm gonna skip the next slide and just talk about, move on for the rest of my time talking about what we do, and then very briefly about ISI and ISD. And so we think about developing these tools to make these compounds. And so we think a lot about mimicking the form of biology with synthetic chemistry. And so in other words, how do we make complex molecules that have function, but how do we do that in a lab? And how do we do it simply, quickly, and with our ability to change it a lot? Because it turns out that a sea sponge in the bottom of the ocean is not undergoing evolutionary pressure to evolve seed treatments or treatments for human cancer, right? It just so happens that a molecule has an effect, right? But can that be optimized? Can it be improved? Can stability be improved? Can the properties of it be improved? And so we want to spend a lot of time thinking about how we can engineer those molecules to be better. And so to do that, chemists like myself and others are really both an architect and a carpenter of molecules. We both design the molecules and then make them. And the corollary to this is, if you were to think about making a house and you gave a carpenter a pallet of 2x4s, but no tools, right? Hammer, nails, and 2x4s, right? You would end up with things that are some combination of 8-foot by 8-foot squares, right? Because you would have no ability to edit and manipulate the materials that you're using, right? The same is true in the biological world. If you want an inhibitor for an enzyme, if you want a molecule that has certain properties, if you screen what's available and they don't work, you're out of luck, right? What are you going to do? You can't manipulate and change it. Chemists can go in and can, in a very defined, specific, predictable, and characterizable way, edit molecules to be different and new, and therefore have different properties and effects. And so that's what we do, and that's what we think about in my lab. We use all kinds of tools, chemical fume hoods, which are always a fun topic on campus, characterize them with things like NMR and mass spec, and ultimately get these products. And so you might ask yourself, you're a chemist, we got chemists on campus, every university has chemists, companies like BASF have tons of chemists, pharma has tons of chemists. Why don't you all just make all the molecules and get this over already, right? I mean, it's just, we just make everything, then we can solve every problem, right? Well, it turns out this is pretty hard, right? And so this diagram is inside the box is a conceptual representation of all the possible chemical space available to the elements if you were to combine them in somewhat reasonable ways without being too ridiculously big or impossible in terms of the chemistry. So that's your possible thing. The highlighted area shows, or the colored area, I should say, shows what we've made thus far in the course of human history. And the darker red means that's extra density in those areas. And so it shows us a couple of things. One is we made a lot of the same stuff, right? And B, there's a whole lot of white space there. And so it turns out that if 10,000 chemists were to prepare one compound each per second, it would take that, I don't even know what it is, number of years to finish the white space on this slide, right? And so the point is, we're not going to do this, right? And so how do we think about using tools like AI, machine learning? How do we think about overcoming those barriers to more comprehensively cover this white space and to provide some of those solutions that we need for all these different areas? But we need to do something other than just hire more chemists, right? Because, by the way, it takes weeks, sometimes months to make molecules, not a second, right? And so just for some perspective there. And so these kinds of compounds that we make, so here's blockbuster drugs. I could have a similar slide on blockbuster agrochemicals, right? They all look kind of the same if you squint and don't know anything about chemistry, right? They're relatively small. They have these N's and O's and lines, right? You know, but the point is, that's actually the whole point. The whole point is that I could show you 10 blockbuster drugs and I could show you 10 important agrochemicals, and you know, the average person wouldn't be able to tell me the difference, right? And so that's why, from a discovery point of view, if we can create pipelines where we can look at all of these things and not just one of these things, we can create a lot more value out of the chemistries folks like myself are developing. Because we might fall flat on our face trying to cure cancer, but maybe we have an inroad in a fungicide or pesticide or nematicide or whatever it may be. And so the kinds of molecules we work in my group are all over the place in terms of their structure. We work on mainly marine natural products, and so these are molecules that come from sources in the ocean. There's lots of different rhymes and reasons for the molecules we work on. A number of these molecules on the bottom where it says new methods actually have been screened by BASF for a number of different effects. Some of them have some modest effects, but nothing spectacular. And there's a couple projects in the group that actually aren't on these slides that are in collaboration with Corteva, looking at some new natural products that have some interesting agro relevance. And so, you know, as part of our group, a lot of the interest has been in infectious disease. And without getting, into that, I'm just going to fast forward through that part. Really, we're thinking a lot about, treating microbes, killing microbes, thinking about biofilms and modulating biofilms. We found molecules that both kill biofilms, that stimulate biofilms, that have all kinds of effects on that world. And that's been a major focus of our lab over the years. And I think just to show you some of those compounds, and so the lipoxyzolonone, leopolic acid, and synoxyzolonone, these are molecules that are natural products that were sort of, early in my time here, but have now become quite translationally relevant. And we actually have a startup company developing the compound class on the left here as human therapeutic antibiotics. And so that's been a more recent development. Ultimately, I'm just going to fast forward through this for time, but that's come to this company here, Synox, that I just mentioned. These molecules are actually fatty acid biosynthesis inhibitors. It turns out that's a relevant target, not just in microbes, but also in other targets, including plants. And so we're interested in investigating that as well. So for my last couple minutes, I just wanted to introduce this crowd to, I guess, the next PSB, if you will. And so that's going to be the Integrative Sciences building on NC State's North Campus. And the focus of this is, really thinking about, PSB is this hub of uniting plant sciences. ISB is going to be a hub for uniting people that like to think about molecules and think about, I think as BASF would say, to think about chemistries. And, so that's really the main goal of this. It's on a memorable site for anybody who's spent time on campus where Harrelson Hall used to be. That was demolished for this permanent looking garden during my time here. But it's now the site of construction. This is actually a couple weeks old now. That this is the topping out should be the last part of May. And so we're getting there. And so it's the building is coming up, and in sometime in 2026 will look like this. And so it'll be right on the Brickyard, right where Harrelson stood. The most important feature is it completely blocks your view of the surrounding buildings from the Brickyard, and so you no longer have to see them, which is I think a strength for campus. but seriously, this building, we really have three major buckets of impacts. I think several of them very much align with the goals of PSB and PSI. I think the one area that's a bit different for integrative sciences is we will have a major curriculum and education component for undergraduates in the building. And so the bottom two floors of the building will be very focused on education, particularly lab-based education, and providing interdisciplinary, integrative education across chemistry, biology, biochemistry, physics, math, in these labs that are going to be inquiry-based and problem-based and not cookbook and yeah, I'll save the rest of the comments for some other time. But that's the vision. The research areas of most interest, the molecular biomimicry I already mentioned. What I didn't mention is that also certainly includes synthetic biology and aspects of that. Molecular metabolism, largely driven by our biochemistry department, molecular and structural biochemistry, as we were talking earlier, to be more specific, and then accelerated and predictive technologies. Is an area that we're super excited about, leveraging a long history of our engineering programs, but really now interfacing them with chemistry. I mentioned this sort of accelerated platform for discovery. We've talked with a number of partners about this, not necessarily in the seed treatment space, but in the just the molecule space more broadly, and really thinking about not only how can we use this technology, but how can we push the boundaries of this technology in collaboration with partners so that they can use it themselves, so that we can use it, so that everybody can have these kinds of platforms to use in their discovery pipelines. And so we're hoping to create a consortium with NC State as a leader in this space, and that should be an emerging area. I care a lot about innovation and partnerships, and so both in the education and research front. And so, one of the things I'll just leave you with is that we are really excited to think about how we can partner with industry in what we teach and how we teach it and what we do in advanced labs and why we do it. And then ultimately, of course, in research projects and other applied areas of science. But that whole spectrum is an area, and we want to work together with PSB and PSI in thinking about where those synergies are, where there are molecular interests and plant science interests, and can do that collectively. Our goal is certainly not duplicate or compete, it's to enrich and lift up. And so we hope to be able to do that. And so we're really excited about this and thinking about moving this forward. As you saw, the building is in progress and we're building lots of pieces underneath that. There's lots of folks from BASF involved in this already. Happy to have more. That's an ongoing area for collaboration. Just briefly, I want to thank my research team because any of the stuff I briefly alluded to, I did none of, and I'm really thankful to them, and be happy to answer any questions anyone has in terms of either what we do or ISB, or we can continue this conversation upstairs, and because I know I'm out of time. Thank you.
Questions?
Where do you come up with the names, the chemical names that you see out there?
Yeah, so those are coined by the people that isolate them, which unfortunately is me. I missed a calling in life, I think, because when you're a natural product isolation person, you get to go to like Fiji and scuba dive and get these sponges, and then the rest of it isn't so fun actually, but that part's fun. They're usually named after either the location that they're found or the organism that produces them or some combination. Like a molecule we're working on is called abyssamycin, and it's called a mycin because of its origin in terms of how the organism that forms it, but it's called abyssamycin because it was found in the very deep seas, and so it was from the abyss. Right. And so there's a little bit of creativity and a little bit of science in it. The more complicated part is when multiple people isolate the same thing and call it different things. That's a little bit more complicated. Yeah. Yeah.
I think it was the slide that had your startup company on it.
Yeah.
There was saw MRSA on there. Is that a key focus to develop new classes of antibiotics for that kind of action?
Yeah, so yeah, this project, so the origin of this is, so you have gram-positive bacteria and gram-negative. gram-negative are harder to treat, but gram-positive are very challenging to treat when they form biofilms. And so any kind of implant infection, hip infections, heart infections, catheters, the challenge with those infections is that they have biofilms, which are the same things you see in your shower and your sink, that sort of growth of bacteria on there. And so none of the existing FDA treatments for antibiotics actually address biofilms. And so if you have a hip implant and it gets infected, the standard of care is to surgically remove it, stuff it full of some sponges soaked with antibiotics, wait, and then try it again. It's incredibly invasive and expensive. And so we're trying to develop a single agent that can treat both planktonic bacteria, the ones that are floating around, and ones that have laid down in these biofilms. Yeah.
Sound like a really promising molecule. How long and what is the process after that then, like, to basically get it to become a product?
Yeah,
I kind of know
similar.
a little bit, like,
Absolutely. The comment earlier was maybe it takes 12, 13 years. Similar. Probably more like $1 billion. And the thing is, for everyone that makes it, a number of them fail very late stage. And so it's not 1 billion, it's probably 5 billion collectively. And so, it takes a long time and it's very expensive. It depends on what you're treating. Cancer is hard, but clinically it's a little bit easier because you give people something, give something else, don't respond, right? And then, I'm going to try this new experimental thing, and if it doesn't work, you're going to not make it anyhow, right? But infectious disease doesn't work like that, right? You can't have somebody that presents an infection in the clinic and then say, well, I'm going to try this new thing, good luck, right? It's because if you have things that could work, right? And the problem is with infectious disease, if you have it and you can't treat it, you're going to be dead in a few days, right? Cancer is a bit slower, and so you have the time to undergo that. So there's challenges in there. Talk about that for hours, but it takes about at least a decade, and costs a whole lot of money.
Also involve like
clinical trials, phase 1, 2, and 3 clinical trials. That's where most of the expense is. And so however much money we could possibly thinking about spending on the stuff we do, it's pennies compared to how much the clinical trials are going to cost. Yeah.
One more question.
AJ, that's one.
This may be an over the glass of wine question, so we can continue this upstairs. But
Thank you.
but my question was around drug design.
Yeah.
And the ability and where we are in kind of state of play to apply these machine learning AI tools for that, and not only to perhaps find an efficacious molecule, but at the same time look for a molecule that has low toxicity, low environmental impact, low cost of goods. Are we in a kind of space now where we can look for all of those different functions kind of simultaneously?
We are a lot closer than we used to be, and there's some really great stories of success, right? There's also a large number of stories of complete and utter failure. It really depends on what you're trying to do, how much we know about it, and how good the models are, right? And so I think the biggest challenge in that space is that AI and machine learning are only as good as the data sets you provide them. And it turns out that, the language models, ChatGPT is amazing because it can digest the entire literature ever known to man and can then use that to create new, right? Or some combination of old, depending on who you ask. But in the molecule world, AI does not have the ability to process these structures I'm drawing here. We have to provide that in a way that is processable, right? And we have to do that for the entirety of everything ever made. And we have to also have real, valid biology, which oftentimes is not, right? You see some paper, oh, it does this. No, it doesn't, right? And so, but if you give AI something that says it does, now your whole model's wrong, right? And so it's about having chemical data that's correct. It's about having biology data that's accurate and real, and combining those into a system that's big enough, a language model that has millions of data points, to then be able to make predictions that are of meaningful value. And so it's the data curation challenge that is the limitation to be able to do it. If we had that billion data point set for a particular thing you were interested in, I think we could use AI and machine learning to make a drug, and I think it could happen fairly quickly. In most cases, we don't have it. We can talk about public-private partnerships and things to try to make it better. It's complicated. Yeah.
Let's thank both our speakers again. Out of curiosity, can you raise your hand if you're, we'll say, internal NC State in the crowd? And then will anybody who's external, please raise your hand. Awesome. Well, I'm really glad to see a mixture of both sides of academia and industry in the crowd. From here, we're gonna go up to the third floor. You can take the elevator, which is off the main lobby, go up to the third floor, and you're welcome to come join us for some snacks and some drinks and discussions.
Sure.
I don't think so.